Hi, everyone. Welcome to today's Global Grand Rounds. My name is Aisha, and I'll be your host for today. Before we start, I'd like to go over a few things. So this presentation will be recorded and uploaded to our YouTube channel. On Zoom, you may turn on the feature called closed captioning during the presentation to allow for subtitles. Please also create a Figure 1 account if you would like to gain CME credit. And if you have any questions about this, please let me know. I provided all relevant links in the chat, and now I'm going to hand it off to Dr. Pecchia, who will be introducing our host.
Thank you, Aisha, and welcome everyone. Glad to be here again in the next installment of our Global Grand Round series supporting the mission of global offsite care. I'm Gregory Pecchia, physician and volunteer medical director for the organization and also faculty in telehealth and telemedicine at Western University of Health Sciences in Pomona, California. We will have, in fact, some guests today, student members of the class, graduating class for this year at Western University, and we appreciate their attendance and interaction with our speaker. And speaking of which, I'm happy to introduce our speaker today, Dr. Andrius Baskys, who is a physician, psychiatrist, neuroscientist, and educator, and colleague at Western University of Health Sciences with a long-standing interest in applying genomic tools to clinical care. He trained in psychiatry at the University of Toronto with fellowships at McMaster University and UC San Francisco. He has held academic appointments at several institutions, including University of Toronto, University of California, Irvine, and here at Western University of Health Sciences. Dr. Baskys has published over 200 papers, presentations, and reports with contributions in neurodegeneration, psychopharmacology, and pharmacogenetics. His current focus is on integrating pharmacogenomics and next-generation sequencing into real-world clinical decision-making. And with that, I'm honored and happy to introduce Dr. Baskys, speaking today on Precision Medicine Starts Here, integrating clinical genomics into everyday practice. Welcome again, Dr. Baskys.
Thank you very much, Dr. Pecchia, and thank you all of your team and for preparing this and for the opportunity to present here and for this great introduction. By way of disclosure, for the CME purposes, I am a founder of MoodNote LLC, which is a company dedicated to software development and data analysis. In terms of, I would like to acknowledge also quite a few people who contributed to this presentation, maybe not quite directly. These are my great students they used to be at Western U, now they moved on and they are in places like UCLA and other places doing their programs. I also appreciate internal grant support from Western U that allows us to conduct genomic analysis. I am indebted to the NIH and specifically dbGaP program that allows us to download the massive amounts of data for analysis. I thank all the administrators that usually provide me with time and ability to run my experimentation, and also Dr. Barnes and Dr. Magoon for the insightful clinical discussions. I was asked to prepare something funny from my life to connect with the audience, and I was thinking that I couldn't find anything funny about my life to share with you, but just today it occurred to me that the probably funniest part of it is that I'm a psychiatrist. I'm a board-certified psychiatrist. Today I'm going to talk about things that have nothing to do with psychiatry, which is cardiology and nephrology. I'll present you two cases, and I think since I do not know these fields, so it might sound funny, I hope. So starting with about genomics, and we need to refresh the concept of a gene, which has been coined by, I don't know if it's been coined, but it's conceptualized by Erwin Schrödinger, a physicist in around 1944, who thought that living organisms cannot transmit information easily from one generation to another, because that information will be destroyed by Brownian movements. Therefore, he suggested that information that is being transmitted needs to be coded in some kind of way. And that's what the gene does. Now, in terms of genomics, that's a study of the organism's entire genetic material, including all of the genes, all DNA sequences, and how they interact to influence the organism's traits and characteristics. Now, in terms of clinical genomics, that's a genomic data-guided diagnosis and treatment. It has several names: genomic medicine, precision medicine, clinical genomic. In a nutshell, this diagram illustrates how it works. We see the patient as a data set obtained through the next generation sequencing. That data can be processed using a lot of bioinformatics tools, and the diagnosis comes out. That's in a very simplistic way, but that's how it is. Now, for those who are not familiar with next generation sequencing, that's a term used to describe modern sequencing technologies. Here is a sequencer on the right. This is a short video I recorded in Hudson Alpha Clinical Service Labs in Huntsville, Alabama. So it looks, we were touring it, and it looks a little bit like a dishwasher, and that's one of the Luminous sequencing machines. Just a little bit, a few terms to keep in mind. The genome contains about 22,000 genes, the human genome. There's about 3 billion letters. It's like a text. And that's most remarkably that 99.9% of humans are basically identical. It's just a little bit, about 0.1% of a genome where variation occurs. So this variation that is important for us, the hope is that since these variants are associated with disease or how they react to different medications, they could tell us something about the disease and help to treat them. So what is exactly a variant? There are different types of variants. The most common variant is a single nucleotide polymorphism, or a SNP. So the diagram illustrates 3 people who all have identical DNA, and in one location, one letter keeps flipping. One person has A, another person has G, and a third person has T. So the address, that location, is known as a SNP, not the letter, the location. There are all kinds of variants. This is the simplest and the most common one. How do we transcribe the data into the variants? This is a picture that illustrates what comes out from next generation sequencing machine. I call it the hanging socks. And so, in fact, we don't sequence DNA. What we do, we break it into pieces. The machine does. We don't do it. These pieces, they're shown here as little gray kind of bricks. They are called reads. They have about 100 letters, and the computer aligns them so that we could see where the actual variants are. Down below here is a diagram of a gene, and as you can see, it's sharp and clear. So these darker parts are the protein coding parts called exomes. Those are the empty parts, usually regulatory regions, they're called introns. And once we align them, we get this kind of fuzzy picture corresponding the protein coding parts. And if we look very carefully here, there are little areas shown in green that vary from read to read. And those areas represent a SNP, which we can then statistically identify and see it. So in this particular gene, we see two variants. Now, the number of these reads that are aligned, it's called the sequencing depth, and it's a very important parameter because the higher the depth, the greater the accuracy. As you can see, the depth is lower here, so nothing really comes out. So this is what costs money, because the more of reads we have to align, the more machine time it takes, the more processing efforts it takes, but it's a greater accuracy, and we need to pay a little bit more for that. So this is a next generation sequencing in a nutshell, or hanging socks. So what do we get out of it? Let's look into some cases. Today I'll present two cases, two clinical cases. One is cardiovascular and the other one is kidney disease. It is quite remarkable that since most of my patients, they come to me with some kind of psychiatric condition, sequencing reveals that they don't really, that they have something else that is of probably greater significance. And so far what we learned that the psychiatric conditions do not come out very well in sequencing data. And this is something for a different presentation. So this particular case is of 65-year-old female of European ancestry, whose major concern was lifelong elevated cholesterol, at times reaching 500 milligrams. And she totally was unresponsive to statin therapy, and attempts to treat with statins would give her severe GI effects, a lot of discomfort, and cholesterol level wouldn't budge. She had some cardiac symptoms, pre-syncope, episodes of pre-syncope. She had some documented calcium depositions in blood vessels seen on CT scan. She is under cardiology follow-up. Statins ineffective, and her cardiologist is reportedly very frustrated with that. She has some other symptoms. She has difficulty walking. She had been diagnosed with hyperparathyroidism, and her family history is remarkable for early cardiovascular death. So this is whole exome sequencing. There's no physical or any kind of other analysis conducted as part of this case. We performed whole exome sequencing using the Tempus laboratories, and we usually what we get is FASTQ files, and we align them, as I already mentioned, and then we look for the variants. In this case, we use a lot of, this analysis involves a lot of databases. Most of them are publicly accessible. I listed them here, and that's not always very easy to work with them because they are very big, and they need to be properly aligned and queried by using software tools. So these are the common databases. It's a SNP database. This is ClinVar. It's a clinical variant database. This is population nomad. It's a population allele frequency database. This is a predictive scores database of various computer predictors. This is gene disease association database. This is UMLS Metathesaurus that allows us to make organ system classifications. And these are common software tools. Free-based is for variant calling. SNPF and SNPSIFT are the tools for the variant annotation, and Intervar is a tool to operationalize American College of Medical Genetics-based classification. What we're looking for is four things. We're looking for variants that have a certain level of clinical significance. We're looking for variants that have very low frequency in the population. We're looking for variants that are computational predictors, predict that they are deleterious, and we're looking for the variants that are in a conserved region of a gene. So these are the four things, four pillars that need to match in order for us to conclude that the variant that we find is indeed deleterious and might be contributing to the symptoms of a disease. This is our general pipeline or workflow. Starts with the patient. We can use blood, we use saliva, and here is a sequencing machine. This is a first file, the hanging socks file that comes out. Then we process these files, we call variants, and then we filter. Filtering is a complex procedure designed through the trial and error, and it's something that we are never happy with, but because we want to improve it all the time. And then there is a literature search, because It requires some kind of a knowledge base to find out what the variant is all about, and we provide the information to the patient. So for this particular case, the key finding was in the gene ApoE. ApoE stands for apolipoprotein E, and this patient had the following change. This is a variant. The variants have addresses. This is the address of a variant. The name of variant starts with RS, and this is a address chromosome 19 and a position 454119141, counting from the beginning of that chromosome. And the patient had 2 Cs in that location, which was a homozygous variant. The ApoE variants are defined, the ApoE genotype is defined by 2 variants, this one and RS7412, and this lady did not have a variant there. As a result, her genotype is translated into what's called ApoE44, and it's considered to be a pathogenic by ClinVar database, which, as I mentioned, is clinical database. So somebody determined that it is disease-causing. It causes protein change, cysteine to arginine at position 130, amino acid 130. And it's relatively common, actually, the C is common in the population. CC is not as common in the population. And CAD is one of the predictors, which is also relatively low. I'll explain you CAD a little bit later. So this is a key finding. And let's see, so what does it mean? And what is that ApoE gene? It's the protein encoded by this gene is a major apoprotein of a chylomicron. It binds to a specific liver and peripheral cell receptor and is essential for the normal catabolism or degradation of triglyceride-rich lipoprotein constituents. And this gene maps to chromosome 19, means that it's in the chromosome 19, and several other genes nearby. And importantly, mutations in this gene result in several diseases. One of them is familial dysbeta-lipoproteinemia or type 3 hyperlipoproteinemia, in which increased plasma cholesterol and triglycerides are often found. And I'm sure that many of you already get the gist where this is going. Just to show you a little bit more in detail, this is what the gene looks like. So this is ApoE gene right here. These are the exons, protein coding parts. These are regulatory regions. Down below here, as you can see, these are the variants that the gene has. And this gene has 2,765 SNPs in it. However, a small proportion of them, shown here, are known to be deleterious, and I marked here as red lollipops. Another proportion of SNPs are known to be benign, meaning that they don't cause any disease. And then there is a group of variants marked here in yellow, and those are called variants of unknown significance, or VUS. So these are all the variants that we know about. So there's a lot of variants we know nothing about, but there's a small proportion that we know something about. And if we look again, this is ApoE gene. We can look at it again, the diseases. I put this kind of ugly looking screenshot to show you that this is how we see the data, because most of bioinformatics processing uses a very simple tools that are usually displayed on an Apple computer terminal app. It's not very nice internet depiction or description of what we do. There's a variety of diseases, lipoprotein-related diseases, and also dementias, Alzheimer's and Lewy body, macular degeneration. So these are diseases that are linked with ApoE gene. So getting back to the variants that we found, so the definition of the genotype arises from 2 variants, as I already mentioned, this 429358 and 7412. And in this situation, it's a simple formula to calculate it, but just for the sake of complexity, I'll ask you to believe me that this is correct. It's a CC homozygote variant. And the other variant, another location is TT homozygote reference. So there is no variant in that other location. So that gives us ApoE44 genotype. Let's see what are these things linked to. So the very important part of carrying ApoE4, ApoE genotype is lipid regulation. And ApoE44 is known to convey impaired clearance of LDL and remnant particles of chylomicrons because of inefficient ApoE4 binding to hepatic receptors. It is also associated with neurological risk. Individuals who carry 44 have about 10 to up to sometimes 35 times increased risk of late onset of Alzheimer's disease. In fact, late onset of Alzheimer's disease is probably incorrect to say because it starts much earlier for the carriers of ApoE44 compared to ApoE33, which is a majority of the population on Earth. Remarkably, carrying ApoE44 affects the drug response. And the first thing that you may notice there that the lipid-lowering efficacy with statins is either poor or non-existent. And there are some other changes, increased sensitivity to warfarin. And for those who develop Alzheimer's, medications like donepezil tend not to work. How does that link to our patient? We have statin resistance. That's one of the major complaints of a presenting complaint. Vascular calcification, that was another complaint. We do not have cognitive complaints at this point. That's a clinical link of our finding. Now, in terms of treatments, looking through the databases, the treatments would be not non-statin cholesterol-lowering agents for that, like PCSK9 inhibitors. Some nutraceuticals might be useful, such as omega-3 fatty acids and CoQ10, acetylcarnitine. Those would be important. And, of course, lifestyle changes, including Mediterranean diet, regular aerobic activity, minimizing sugars, refined carbs. These are known things. But for this patient, this could be of maybe added significance because lowering elevated cholesterol with statins is not going to work. As I mentioned, that ApoE gene has several genotypes. Those are known as E2, E3, and E4. So total, there are about 6 combinations arising from these 2 SNPs, 2 variants. And the frequency of these genotypes varies across populations. And they have several other clinical roles in addition to the neurodegenerative diseases such as Alzheimer's or conditions such as hyperlipidemia. So first of all, I just would like to talk a little bit more about different genotypes. So E2 is very rare. And it is found in about 1% of the global population, and it's known to be as protective against Alzheimer's disease. E2, E3, it's a little bit more common, and it's found in 10 to 15% of the general population, and it's somewhat protective against Alzheimer's disease compared to E3. So E3 is the genotype that is found in most people. About 60% of the population carry this genotype, and it has what we could say a neutral risk of Alzheimer's disease, a baseline for comparing with other genotypes. Now, there could be a rare variant such as E2, E4. E4 increases the risk, E2 decreases. This would be an intermediate risk, something a little bit maybe higher than E3, E3, but not as bad as E3, E4, or E4. And E3, E4 carries an increased risk, about 3 times greater risk of Alzheimer's compared to E3. And that genotype is found in about 20 to 25% of the general population. Now, E4, found a typo here, very high risk, is found in about 1 to 2% of global population, and it can carry a very high risk of Alzheimer's compared to most people E3 carrying E3. So that's what our patient has. In addition to Alzheimer's disease, the ApoE genotypes, the ones that I just mentioned, especially those involving E4 allele, are strongly associated with lipid abnormalities. Those abnormalities are elevated cholesterol levels and poor response to statins. This relationship has several significant clinical implications. And the most important implication is that they exhibit high low-density lipoprotein cholesterol levels. And the statins, it's the first-line treatments for hyperlipidemia, are less effective or completely ineffective in lowering cholesterol in these patients. As a result, physicians unfamiliar with ApoE genotypes may become frustrated when statins fail to achieve target cholesterol levels. Alternative treatments such as PCSK9 inhibitors, which are expensive, or dietary modifications may be more effective in cases like this one. How does that pertain to personalized medicine? Testing for ApoE genotype and finding out what does the patient have, can guide treatment decisions for both Alzheimer's disease prevention and for lipid management. For example, a patient with E4 containing genotype and high cholesterol may benefit from early intervention with non-statin therapies, as opposed to early intervention with statin-based therapies, which would be totally ineffective. Similarly, knowing a patient's ApoE status can help stratify Alzheimer's disease risk and prioritize lifestyle intervention, for example, such as cognitive stimulation, diet, and exercise. Those are all known to be good protective factors against Alzheimer's disease. As a result, understanding the ApoE genotypes, a role in neurodegenerative and cardiovascular conditions, will allow physicians to provide more personalized care, reduce frustration of patients and their own frustration, and improve patient satisfaction. There are broader implications of ApoE genotype, broader than Alzheimer's and lipid metabolism. And emerging research has highlighted the ApoE role in various other conditions, from differential responses to therapies like hormone replacement therapy, to susceptibility and outcomes related to novel treatments for Alzheimer's and even global health interventions. Let's take a closer look. New Alzheimer's drugs and ApoE4 genotypes. Many of you may know that just recently the 2 new drugs for Alzheimer's disease have been released. I think one just as recently as last summer, and they are known as lecanemab and Kisunla. And one is Kisunla, I think, is by Lilly, and lecanemab, I can't quite remember who produced it. And these drugs target amyloid beta plaques in the brain. So it targets what is believed to be causing Alzheimer's related changes. However, there's a problem with these drugs. The effectiveness, and particularly the safety profiles, are heavily influenced by the patient's ApoE genotype. ApoE E4 carriers, especially those with 2 E4s, are at significantly high risk of developing what's called ARIA, amyloid-related imaging abnormalities, which in simpler words is a condition involving brain swelling or microbleeds as a side effects of these therapies. So naturally, that kind of limits the use of these medications, and many patients may be reluctant to start the treatment, and many prescribers, physicians who treat these patients, may be reluctant to initiate treatments without knowing what is the patient's ApoE genotype. Understanding the ApoE genotype of a particular patient can help clinicians weigh the risks and benefits of these drugs and monitor side effects more closely. Hormone replacement therapy is another clinical field where ApoE genotype becomes important. ApoE genotype plays a crucial role in how women, and this is in reference to postmenopausal or menopausal women, how they respond to hormone replacement therapy. Specifically, women who carry E4 allele are less likely to respond favorably to HRT. There are studies suggesting that ApoE E4 carriers may experience diminished cognitive protection from HRT, and in some cases, increased risk of adverse outcomes such as uterine cancer. On the other hand, ApoE E3 acts as protective genotype, as in women with E3 tend to respond better to HRT with fewer associated risks. Therefore, knowing the patient's ApoE genotype could guide decisions around HRT for menopausal and postmenopausal women. Important. COVID-19. This pertains to the global health measures. The ApoE gene has been implicated in the response to lipid nanoparticles, including those used in Pfizer's modified RNA-based products. The modified RNA therapeutics use lipid nanoparticles, often known as LNPs, that are coated with cholesterol molecules, and that they use ApoE as a shuttle to deliver genetic material into human cells. So ApoE protein facilitate the entry of these nanoparticles into cells by binding to lipid receptors. However, individuals with ApoE E4 genotype lack the same cellular uptake capability. As I mentioned already, that ApoE4 does not bind to these receptors and potentially can reduce modified RNA cellular entry in this group of patients. Approximately 25% of the general population carries at least one ApoE4 allele, meaning that a significant portion of individuals may not derive optimal benefit from modified RNA products due to reduced LNP lipid nanoparticle uptake. Despite this, vaccination mandates, public health policies often did not account for genetic variability, leading to a one-size-fits-all approach that may not be equally effective across all genotypes. To summarize it, the clinical relevance of ApoE genotype is broader than cardiovascular and neurological. ApoE plays diverse roles, and together these roles underscore the importance of genetic testing in personalized medicine. By understanding ApoE's impact on various conditions and treatments, clinicians can use ApoE genotyping to guide the use of monoclonal antibody therapies, mitigating such risks like ARIA in ApoE4 carriers. They could use them for hormone replacement therapy. Genotyping could help identify women at higher risk for adverse outcomes, such as uterine cancer. The impact of ApoE on modified RNA response suggests a need for more nuanced public health strategies that consider genetic variability rather than blanket approaches, and continued exploration of ApoE's role in drug delivery system, cancer risk, and other conditions could lead to innovative treatment and interventions. So in conclusion, the ApoE genotype remains one of the most clinically significant discovery of genetic factors in human health. Its influence on Alzheimer's therapies, hormonal treatments, lipid metabolism, responses to modified RNA technologies underscores the importance of integrating genetic data into both individual care and public health strategies. By tailoring interventions to genetic profiles, the medical community can ensure more effective outcomes across very diverse populations. Let's look at another case, and this is a genomic analysis of rare deleterious CoQ8b variant in a 72-year-old patient with a kidney disease. So this is a case, a 72-year-old female with a history of chronic kidney disease and depression, and that's what I was treating her for. Controlled hypertension, muscle pain, type 2 diabetes, family history of death from kidney disease, underwent clinical evaluation and whole exome sequencing. Bioinformatics analysis conducted using the standard protocols, as I already described, revealed a what's called nonsense heterozygous variant in chromosome 19, resulting in amino acid change and truncation in the gene CoQ8b. Let's take a look at what that means. CoQ8b is implicated in steroid-resistant nephrotic syndrome, or nephrotic syndrome type 9, which occurs due to primary coenzyme CoQ10 deficiency. And what that gene does, it plays a crucial role in coenzyme Q, also known as ubiquinone in some literature, biosynthesis. So it functions as a kinase that is important for aerobic cellular respiration and facilitates electron transport within mitochondria. And it is involved in kidney function. It's involved in podocyte migration and maintenance of kidney function. Clinical manifestations of this mutation presents usually a characteristic of edema, hypoalbuminemia, fatigue, myopathy, and focal glomerulosclerosis, and proteinuria. Importantly, the condition is resistant to therapy. Specifically, it poorly responds to corticosteroids. We did the same thing as we did with the other case. We performed whole exome sequencing. Tempus Laboratories, it's a CLIA and CAP certified facility, got our data in FASTQ format, aligned, produced and called variants, went through hanging sock stage, performed annotation and filtering, went through all the databases, identified allele frequency, conservation, predictor values, and we used various tools to make sure that which of the variants meet American College of Medical Genetics pathogenicity criteria. We used a variety of data sources, and what we found was a amino acid change due to the stop gained mutation in the gene CoQ8b. Genes have several names. There is another name, ADCK4. Looking into that variant, the protein is shown here, looking in the variant, it's extremely rare. This lady was of African-American ancestry. It's a little bit less rare, but still quite rare, given that we usually cut off our allele frequency at 0.05%. So this was much more rare. And so that was a significant finding indicating potential pathogenicity of that variant. We went through all four pillars of our pathogenicity determination. So this is a variant, truncating variant. The star indicates that the variant was truncating, so the gene could not be fully translated. So we use software tools known as predictors. They calculate scores. We use about 15 predictors. We here shown only two common predictors, CAD and mutation taster. Both gave a deleterious prediction. Allele frequency, as I already mentioned, was extremely rare. Why is that important? It is important because the rarer the variant, that means the more deleterious it is, because most patients die. They can't survive. So those who survive, there are very few of them. So that if a variant is very rare, then that tells us that's not a very good variant to have. Then the conservation. Conservation is important in the context that the more conserved region of a gene, the more likely variant will cause disruption. and the American College of Medical Genetics prediction were also likely pathogenic. What we did then, we calculated the impact of that variant, all the variants, on different organ systems. We use that using a DisGenet database, which allows to calculate gene disease association rate. And gene disease association rate is shown here on the y-axis, goes from 0 to 1. And so some genes are very highly associated with the disease. Others are less highly. The majority of variants that we find are in the genes that are not that much associated with diseases. And we showed not the actual diseases, but we showed organ systems, how these genes associate with particular organ systems. So this is our variant, was in the gene CoQ8B, and this is associated with genitourinary system. And it's pretty high. Usually we see somewhere around 0.6 and above, the patient usually has a disease if a patient carries a variant associated with that disease. And I want to point out that, yes, there are some other variants, for example, associated with cancer, but the patient does not have a cancer. We can only talk about the diseases that patient has, not the diseases that the patient does not have. There are other diseases, other kind of endocrine-related variants, which we do not know what the significance of these variants is, but it was important finding that our variant was with a gene that is highly associated with genitourinary organ system. So the inheritance of that gene is not fully characterized, but is classified as 50-50. And sometimes the portions of a gene could be inherited in a dominant way, and the portions in a recessive way. When it's a recessive, so a mutation can have either very small effect or no effect, or can have a full effect, which is another mutation, whereas if there is another mutation that also impacts that same gene. The variant is unknown, so the specific impact on that person's phenotype is not known. We speculate that this is important because, as I showed you, that some variants are known, but others are not known really that well in relation to a particular disease. And as I mentioned, there are some other genes that also could be linked to the similar presentation, which we do not know what the role is at this time. But what we did actually with this lady, we looked at the literature and they found that taking CoQ10 supplementation in a variety of diseases, including kidney diseases, is helpful. At a dose of 400 milligrams per day, there are no known side effects of CoQ10, and the product is available over the counter. So what we did, we performed this sort of observation of our patient. We recommended with the input from nephrology, to take CoQ10 supplementation for about a year. And what was being measured is microalbumin versus creatinine ratio. And that's a standard metric for nephropathies of this kind. And where she started it a little bit more than a year ago, these were the values here, which pretty quickly dropped and remains not fully normal, but somewhat lower than they were initially, indicating that there might be at least a partial benefit of CoQ10 supplementation for this patient carrying mutations that impact CoQ10 production in the kidneys and contributes to the kidney condition on the nephropathy. So these findings emphasize the importance of assessing CoQ10 deficiency, considering supplementation to potentially help improve kidney function. And it's CoQ10 supplementation is actually quite readily available. Not always very cheap, but it's available over the counter if needed. And in conclusion, to finalize, I just want to share some final thoughts, future directions. And the one conclusion that comes out of this is that whole exome sequencing enables precise diagnosis and maybe tailored treatment for patients carrying particular mutations. There are caveats, of course. The data is probabilistic, not always definitive, and that's what most physicians have a hard time understanding it. The uncertain significance of variants, it's still quite common. There's a lot of variants that are of uncertain significance. Therefore, the clinical correlation is paramount. What should we do in the future? What I would like to see happening in the future, I would like to see physicians being trained in bioinformatics, at least in fundamentals, so such as learning to work with databases, ClinVar, Nomad, DisGenet, and so on, and to facilitate the interpretation of genomic outputs. I often see clinicians receive genetic data totally lost without not being able to interpret the data in any meaningful way. Interdisciplinary collaboration is important. Clinicians, geneticists, bioinformaticians need to work together. Machine learning models important, because without the machine learning models, LLMs, we could not do anything here. And developing algorithmic pipelines is a critical step in the future. So that concludes my presentation. If anyone is looking for clinical genomics evaluation or second opinion, please contact me. This is my Western U email, and telemedicine appointments available, so you may be at any part of the world. We do have some grant money, so in some cases we could help out to offset the sequencing costs, because sequencing we have to pay for the machine, so that's the major costs. And for more information, please visit my website, moodnote.com. And we still have a few minutes, right? So please go ahead and list your questions if you have any.
Dr. Baskys, thank you so much for that.
Okay.
And with the few minutes we have available, open to questions. You can either post those in the chat or just speak up, unmute yourself, and pose your question.
Yeah, just speak up because I don't know where the chat is, frankly.
So I'd like to break the ice with a question
Go
about
ahead.
basically the value proposition of this incredible technology from the perspective of the public health kind of population health perspective. And let's start with a focus question on case number 2. As you mentioned, you were seeing the patient for depression.
That's correct.
Is that right?
Very mild depression.
Okay, but that was what brought the patient.
That's what brought the patient, yes.
Yeah. So what was the trigger for sequencing that patient? So in everyday practice of psychiatry, for patients who are being seen like this for mild depression, what would the indication be? What should pop up in the mind of the treating clinician, in this case, the psychiatrist, to actually do the sequencing?
Yeah, that's actually a very good question. Thank you, Gregory. The trigger was the pharmacogenetics. In my clinic, before prescribing medications for depression or something else, we usually perform a pharmacogenetic analysis to identify the most optimal selection of medications. And the laboratory that was providing pharmacogenetic data also provided whole exome sequencing data as well. And that's how we got that data. Does that make any sense?
Almost in a strange way, that almost like an accident in the genetic kitchen.
It's a genetic kitchen. Yeah, that's how that laboratory worked, basically. They used a high-end technology to answer a simple question. Most laboratories don't do it. And basically, that's how we got the data. So as a result, almost every patient that was coming to my clinic and was started on some kind of medication got their genome sequenced.
Wonderful.
Yeah. The laboratory was on an unsustainable path, though. They don't do that anymore.
Thank you for that. Yeah, and really on that, while we're awaiting any other questions, staying on that same theme, at what point does the cost of screening, and anyone, quite frankly, for treating anyone, let's say using the same model, I'm going to treat someone for hyperlipidemia, let's say, and I haven't even gone down the path yet of knowing whether or not they have a genetically based hyperlipidemia, but I'm going to test them because I want to be sure I don't prescribe the wrong agent. So my thinking in, let's say, primary care is, gosh, the testing is affordable enough at this point that everyone that I'm going to treat with just about any medication, I'm going to perhaps want to know about what the prediction is, whether or not I should lower the dose, increase the dose, or avoid use of a first-line medication that I might have. So how is that looking in terms of the value proposition, in terms of the cost of screening?
For the simple pharmacogenetics, which is not based on sequencing by most laboratories, based on PCR, the major healthcare providers, such as Medicare and probably Medi-Cal, they pay for it. So that cost, if a patient is a Medicare beneficiary, they don't pay for it. So it's accessible to everyone. It's a little bit more complicated with commercial insurers, and that depends, as the commercial insurers have different levels of what they pay for and what they don't pay for. But typically, commercial insurers do not pay about the first $300, which is roughly of how much the test costs.
Well, great. Unless we have any other questions, comments, I'm looking in the chat. See some thank yous there, and I appreciate that very much. And I want to thank you, Dr. Baskys, for being with us today and sharing this important information. By the way, I see a little typo for those who are going to be emailing you at your email address.
Letter U is missing.
There we go. So it should be at Western U.
That's my ADHD is probably acting up again. I'll have to address that in the therapy session.
Maybe we should sequence you. Maybe that's a symptom.
I've done it, actually. I sequenced myself twice, and I also sequenced at least 2 of my children and 1 grandchild.
Great. Thank you again for sharing with us. We are short on time. And with that, I will hand it back to Aisha. Thank you, Aisha, for leading today. And again, also to all of our volunteers at Global Offsite Care for their hard work and for supporting this monthly series. Aisha, back to you.
Thank you, Dr. Baskys, for your insightful presentation. We are almost done today, so this recording is going to be uploaded to our YouTube channel in a couple of days. And as a last reminder, if you could use the link below in the chat box to create a Figure 1 account for CME credit. If you're interested, please sign up to receive our monthly Global Grand Rounds invitation. Our next Global Grand Round is scheduled for May 14th, and as always, we appreciate your participation and continued interest in Global Grand Rounds, and have a great rest of your day, and we hope to see you guys again.